thanks so much for coming, and also thanks so much to the organizers for inviting me to give this talk. My talk is entitled Identification of Feedback Controllers in Locomoting Animals. And my lab has studied a number of locomoting animals over the last 10 or so years, starting with the lowly cockroach, Paraplaneta americana, shown here running along a wall. This is work I did with my postdoc supervisor, Bob Full. And I continue that work to this day, and also some work all the way up to quote unquote humans. Some work that we've got going with Amy Bastian on running on a split belt treadmill. While I was on sabbatical last year, I forayed into some other bugs. Here's a hawk moth on a tethered preparation where we're doing visuomotor abdominal reflex system identification and trying to understand the role that this gigantic abdomen that these moths have, what role that might play in stabilizing flight, and we've got some really interesting findings that we're in the process of publishing on that. Some other work that I did with my graduate student, Itai Roth, while we were at University of Washington last year. There's a little fly tethered right there to the end of that sting, and the fly is sitting inside of a visual arena, and we move the stripes around and measure the torques. This is work mostly done by Dickinson's lab, but we collaborated with him on some system identification work there. But today what I'm going to tell you about mostly is the work that I've done at Johns Hopkins with my collaborator Eric Fortune on the weakly electric glass knifefish. And so I hope you see some of the things that we've been able to do with the system identification approach to this animal locomotion, but I want to sort of start by stressing why system identification of fish, as opposed to, say, engineered systems, is difficult. First of all, behaviors vary greatly between individuals and even within an individual, depending on a number of factors that are not necessarily easy to control. And also, animals exhibit a broad variety of behaviors, and it's not often easy to tell which behavior they're performing at any given moment, and they can be performing multiple behaviors at the same time, and they can switch behaviors during an individual trial. And so how we've addressed this is by tackling a set of problems that we've just borrowed some terminology, smooth pursuit behaviors. So smooth pursuit eye movements are ones in which if I move my hand in front of your face like this and ask you to track my hand with your eyes will track my finger and stabilize the visual image of my finger on your retina. And so that notion of smooth pursuit, which was used for those kinds of tasks, we borrowed for certain kinds of locomotor behaviors where you're trying to regulate some sensory value to some equilibrium, whether it's the distance of the cockroach to the wall measured by its antenna, or the position of a fish relative to objects in its environment. And so the reason I like these behaviors is, one, they're not uncommon. They're actually all over the animal kingdom. As I've just hopefully showed by the previous slide of just the animals that I've looked at, they can be repeatedly and robustly elicited in experiments. And if you want to do system ID, you need crap loads of data, and so you need to be able to do lots of experiments and get them to do the behavior over and over again. And the other thing is that you can use the data itself to somehow tell whether or not they're doing the behavior that you want them to do. And I think those things are really important. And so here's the glass knifefish, Eigenmania virescens. It's called the glass knifefish because it's shaped basically like a knife, and you can't tell from this picture, but it's semi-transparent. Well, first let me say what's sort of expected about these animals. They have eyes. So here's their little eyes in there. They can see the objects around them like you and I can, but they also have this really interesting electrosense. So they generate an electric field in their tail. That electric field envelops the entire animal, and then small objects in their surrounding modify the electric field. They have different impedance, modify the electric field, and that creates an electric image on the surface of the skin. And so they have this ability to translate the three-dimensional world onto two separate and independent sensory sheets, right? And so they can measure their position relative to the environment in a way that I don't think very many animals can do, and it makes them a good model, therefore, for multisensory integration. And then also, they're really good locomotors, and they have a very, very peculiar style of locomotion that takes advantage of this ribbon fin. So I'll be talking about that as well today, too. So, but actually, the most important feature of these animals is that they like to swim inside little tubes. And so here's an electric fish swimming inside of a tube. It's a different species, but they really like to do this. You just walk into the lab any given day, there's all these little PVC pipes, and you pick it up and you move it back and forth. Inside this aeration tube, there's a fish, you can't quite see it, so it nose dives into there. They just really like to hide in little tubes, and please don't ask me why, we can talk about that over beer. Because as a system ID person, I don't actually care. I want to know the mechanisms for sensory motor control. The question about why they decided to do that particular behavior is certainly interesting, but in the context of understanding the mechanisms for sensory motor feedback control, I don't need to know. So I'm going to break my talk into 3 parts. One is sort of identifying the plant model of the feedback control system. The next is putting the animal in closed loop by basically having my colleague move his hand back and forth. No, actually we use an actuator, but the same idea. And then lastly, we look at, we're trying to now tease apart, this is the most interesting feature of these animals, as I mentioned, I think for understanding feedback control, try to tease apart the separate contributions of visual and electrosensory information. So I'll start with the work done by my graduate student, Shaheen Safadi, on counter-propagating waves in these electric fish. And so this is sort of a simulate animation of that. You can see, I hope you can see in the back there, there's sort of these 2 counter-propagating waves moving together and meeting at the center of the animal. And the question is, what exactly are they doing? And so, and what advantages might that behavior have? So here is an animal, and I'll start by showing it hovering in no flow. So the animal's in a flow tunnel, and in the top video, it's just sitting still. As it sits still hovering, it actually sort of wastes some energy by having these counter-propagating waves meet at the center, as opposed to, say, just lying on the bottom of the tank, which some different species with very similar morphology do. So there's lots of different strategies that these animals could take. But this particular species likes to hover through this active generation of canceling forces. And as you increase the flow speed, the position that those 2 waves meet, which we term for, please give me a better name for this, we term the nodal position, moves toward the back of the fish, as one might expect. There's other strategies that you could have, like changing of the parameters of each half wave, but that's what they do, is they move the meeting point to more toward the back of the fish, and eventually the wave is traveling completely in one direction. Once the flow speed, in this last video, there's water flowing at 12.5 centimeters a second, right to left. And if you increase the flow speed beyond that, no problem, they can swim faster, and they start changing other gait parameters, but I'm not going to talk about that today. So the idea for thinking about counter-propagating waves, so I saw these counter-propagating waves as a possibility for simplifying control, because one of the problems with using traveling waves for generating thrust is that at these Reynolds numbers, to a reasonably good approximation, the force would go something like the traveling wave speed squared relative to whatever the fish speed is. So the problem with that is if you want to change directions, you've got to slow down and stop your fin and then speed it up in the other direction. And that sort of gives you something that in controls engineering we call an actuator deadband. And then when I went to Cornell, I learned about some interesting things that flies do, which is that as they flap their wings and as the animal rotates, they get an extra little drag term for free that looks linear. And so inspired by that, we built a model to try to understand how this works. So here's the ribbon fin, super simple model of it, with waves propagating to the middle. The wave speed is v. The length of half the fish is, say, L, and the speed of the fish is x dot. And if you allow the fish to move this nodal position back and forth, and then build a really, really simple model of what the forces would look like, then what you wind up with is a funny thing where a bunch of square terms drop out, and what you're left with is that the force that acts on the fish due to the interaction between the ribbon fin and the fluid is something like proportional to the change in this nodal position plus a little drag term. And the question, of course, is this sufficient for control? And is this drag force sort of interesting from a controls point of view? Is it big enough from a controls point of view? And so the hypothesis is that they use the change in nodal position to control their thrust, and that this drag term is significant relative to the time scales of locomotion. And so you take that force and you simply make the simplest possible model of an object moving, which is to have force acting on a mass. Then, and since the animal, one of the nice things about this tube behavior is the animal is basically moving in one linear dimension, then we get what I would say is the analogy to the slip model, in that it's the simplest thing we can think of, and it's a mass being pushed on. So the first hypothesis is that the fish uses that one kinematic parameter as the dominant control affordance over its movement, and the other is that passive damping enhances stability. And so when we look over the full range of data, this is the full data set of 5 fish, 3 trials per fish, randomized, all the sort of terrible stuff you have to do with experimental data. This is all the data. So this is the means and the mins and maxes for each fish. So each color is a different fish. The dark bar is the mean, and then the mins and maxes are shown. So very consistently, we see that the one main parameter that changes as a function of flow speed on the x-axis and the different kinematic parameters on the y-axis, the change in length, the wavelengths of the head and tail waves, and the angle of excursion of the fin. None of that stuff changes much. The only thing that seems to change much is where this nodal position is. So that seems to support our basic idea that that's the main kinematic parameter change over this range of flow speeds. But what we don't know, and we don't really want to trust our really simple fluid dynamical model, is how good this damping term is. So we asked our colleagues at Northwestern, my student flew up to work with them for a week or so, to implement counterpropagating waves on the robot and measure the resulting forces. And I don't want to go into the details of all the graphs, but the graphs actually showed that the forces that we estimated using really, really, really simple fluids models actually played out as being pretty accurate in the actual robot. And the bottom line is that when you then do figure out the timescales of locomotion for the animal, that little damping term matters. It has a time constant, the M over B time constant, the mass divided by the damping, turns out to be about 1 to 10 seconds. And the bandwidth of the behavior is about 1 second. So it seems like the damping matters for control. And so, so the conclusion, what we think we understand about the ribbon fin is that the damping term that you get from these counterpropagating waves matters, and that the dominant source of control is the changing of this nodal position. One thing that I didn't mention that we found really interesting is that the 2/2 wave, so there's these two counterpropagating waves, and they don't do this. It turns out they do something more like this. They operate at different frequencies. And in all cases, except for like 3 of our trials, or 8 of our trials out of 150, the tail wave oscillates at a faster frequency than the head wave. And what that really means is that there's functionally separate pattern generators for the 2/2 waves. And if you watch the nodal position, it's kind of amusing. It sort of goes like this, and then it kind of goes up and down. Then it kind of goes like this. It kind of goes up and down as the two waves sort of beat in and out of phase with each other. And so we don't exactly know what's happening. We've tried to get our little EMGs into the muscles right around where the nodal point is, but we haven't been successful. But we find that kind of interesting. So, okay. So the second part of my talk is the full closed loop. So in fact, this is chronologically started much sooner than the understanding of the biomechanics. What we did was we took a PVC pipe, we cut it roughly in half, down the length of it, put it on a boom, put it inside of a tank, put the tube right near the bottom of the tank, and then we use a linear actuator, move the tube back and forth, and then we video it from the bottom. And so this is a bottom view of a fish, and that's the fish, and this is the half cut open pipe. Does that make sense? So, as you see, if I can show this again, so what we do is we think about moving the tube as moving the sensory scene, and then measuring the fish position. So that's this red dot is the movement of the sensory scene. We track that in the video camera. Of course, we know where we're moving the tube as well. And then we track the fish's position, and we basically get the input-output, a phenomenological model of the dynamics between the input and the output. And so what's shown here, this particular data set, this particular trial, is a sum of 13 sinusoids that they're all prime multiples of a base frequency, and the base frequency is 0.05 hertz. So the whole stimulus doesn't repeat until about 20 seconds. And it looks to the naked eye to be a sort of a random signal. And in fact, if you look at it in the velocity domain, it really just looks like noise. And if you look really carefully as a human, you can kind of identify that this peak repeats every 20 seconds or whatever. But it's a pseudo-random stimulus. And that was not my time limit. Okay, good. Whew. I gotta go fast. Okay, so we did that. And I think I accidentally skipped a slide. So the reason I like sinusoidal stimuli for this particular behavior is that we can really tell that the animal is tracking. We know that there aren't sort of extra behaviors going on. I mean, there could be extra behaviors, could be thinking about its mate or something. But for the point of view of the biomechanics, what we see when we take a Fourier transform of the data, this is when you take the raw data of the stimulus and the raw data of the output for one trial and take a fast Fourier transform and plot it in MATLAB, then we see a peak in the blue. The blue is the refuge and the red is the fish. We see a peak at the 13 sine waves. We also see a peak at the same exact 13 frequencies for the fish. So the fish is tracking the tube, no doubt about it. And the other energy in the signal is pretty small. So the main behavior, at least functionally for our purposes, is the tracking behavior. And then we can do other things like coherence analysis, like people like to do in neuroscience, to say that there's sort of strong coherence between the stimulus and the output. But as a controls guy, I like to look at things in sort of magnitude phase space, so a Bode plot. And so a Bode plot is a measure of the input-output. It's a sort of non-parametric way of looking at the input-output dynamics. And so, assuming the system were linear, it's a really good way to look at it. And so the gain and the phase are, the gain is defined as the amplitude of the fish's movement compared to the stimulus movement at that particular frequency. So if the tube moves back and forth 3 centimeters and the fish moves back and forth 2 centimeters, the gain would be 2/3. And then you do that frequency by frequency. And so, for example, when we do a single sinusoidal stimulus, so this is a 0.1 hertz, so it takes 10 seconds for the tube moving back and forth, and I think this might even be at half speed, but the video might be showing at half speed. But anyway, it's a very slow moving stimulus back and forth. We see that the fish do pretty well. Their gain is quite close to 1, and the phase lag relative to the tube is quite close to 0. So perfect tracking would correspond to a line at 1 and a line at 0. And then as you crank up on the frequency of the stimulus, you see the fish begin to lag more behind the tube. So it's sort of, the tube moves and the fish is sort of lagging behind, and its amplitude is attenuated. And then all the way out to very high frequency, which some biologists say, why would you ever want to go out to this high frequency? But their tracking performance is so terrible. But if there's one thing I can also convince you, it's that you have to push the animal sort of far enough beyond, it's boring to get 1-0. You can't really reveal anything about the dynamics when the tracking, when it's doing well. And this is actually one of the problems with identifying very stable closed-loop systems, is you got to push them out kind of beyond their limit. And I feel like we get the most information out here where the fish is kind of doing terrible. And it's 90-degree phase lag, and the gain is down like between 0.1 and 0.2. And in fact, to the naked eye, you can't really even see the fish moving very much. But if you look at the ribbon fin, you see rapid adjustments of this nodal position. And so it's actually doing quite a lot. Oh, and so if you take single sign responses, they should predict, if the system were linear, what the response to a sum of signs would be, and they don't. So the behavior is not linear, and you might think it's naive to assume that a system like this would be linear, but it's a dynamical system operating near an equilibrium point. So there's reasonable expectation that it could be linear. If you look at single sign responses over a variety of magnitudes, they all look the same. And if you look at sum of sign responses over a variety of magnitudes, they all look the same to each other. The difference somehow lies in the structural difference of the stimulus between single and sum of signs. And what we think is that the increased phase lag in sum of signs is because it's harder to predict a sum of sign stimulus. We don't see this when we look at fly and moth data. Incredibly linear when we look at fly and moth data. But for the fish, they're smart enough to go, wait a second, I can tell where that thing's going to go, and it sucks up some of the phase lag in the single sign responses. The weird thing is that the magnitude response is less for single signs at high frequency, but it's only weird until you realize that when you're 90 degrees out of phase, the best thing to do is sit still. Because once you're 90 degrees out of phase, any motion is actually making a tracking error worse. So we think that the decrease in phase lag is some kind of internal estimate or prediction that's helping overcome things like neural delays. And the decreased amplitude at high frequency is that the fish kind of realizes it's sucking at what it's doing, and it sits still, and it does better with less energy. And so I could have said all that with phase plane plots, but I won't. So we saw reduced, I think I just said all this. So I'll just skip all this, because I said it all. So the last thing that I want to talk about, and I don't want to go too much over my 10 minutes, by 20 minutes, and this is brief, is our initial forays into understanding the integration of visual and electrosensory information. And so up to now, we've moved an object, and the animal can see it, and it can sense it with its electrosense. And so we have no way of teasing apart the relative contributions of these two blocks. And, but an interesting observation that Sarah Stamper in our lab explored in a recent paper is that if we start, this is hard to see what's going on in the video, but that thing is the fish, and this is the tube, and this light bulb indicates that the lights are currently on. And it looks just like the data I've shown you before, except it's harder to see, because of the way we had to do our lighting for this trial. And watch what happens when we turn off the light bulb. We see a categorical change in behavior. It's still tracking the tube if you look at the mean response. Similarly, if you go the other direction and start with the light on and turn it off, it ends up being actually even a little bit more dramatic to watch. Categorically different behavior when it's in the dark. And so this is that same data but just plotted against time, and you can see the fish is actually still tracking quite well when it's doing this ancillary sort of probing behavior on top of things. And what we found is that as you increase the conduct, we really think this is active sensing, and our measure for that it wasn't just behavioral or motivational, like, oh, it's in the dark, I'm going to party, is that if we crank up the conductivity by adding salt, they move more. And the reason they need to move more is because when you add too much salt to the water, it sort of shorts out their electric field, and it doesn't work as well. This is our hypothesis, but it's certainly well correlated with this data. And you see the same trend even when the lights are off. So what I'm showing here, I've explained this plot, is just a broad spectrum frequency analysis of this ancillary movement. And it increases in magnitude across spectra when the fish is in the dark. And so lastly, I have one data point more to show, which is that when we do we build a new shuttle that allows us to, it's basically a virtual reality where we project lights onto the back of a translucent tube. And that way we can move, and that's showing you with the ambient lights turned on so you can see both the tube and the little stripe. But when we turn off the ambient lights, all you can see are those little stripes. But the fish can feel the movement of the tube with its electrosense, and it can see the movement of the lights independently of that. And when we do so that allows us to independently move the visual and the electrosensory reference, potentially teasing apart separately the contributions of those two senses. And what we find is that depending on conductivity, they change their sensory weighting to visual and electrosensory information depending on the stimulus details. And so we see contributions to both visual and electrosensory feedback. And so conclusions are slow and boring, so I'll skip them and just get to the interesting thing to say. So we've got feedback control sort of is my approach, and I like black boxes, in this case white boxes with arrows connected to them. There's lots of stuff we ignore. Everybody ignores it. We ignore it too. See? Ignore it. We try to do as many perturbations to the system as we can and measure as many things with our neuroscience collaborators as we can. And then make more boxes, because boxes upon boxes is what control theorists do. And lastly, I want to say that I agree with Chris yesterday saying goal-directed behavior is essential. Transient analysis is essential, and I would argue that goal-directed behavior makes it easy to elicit transients, and then the rest is just speculation that I can ignore, so I can skip. So, thank you. Sorry?
Okay, so we have about 5 minutes for questions.
I'm not sure I understand. So when you move the tube is allows at these Reynolds numbers, there's very little coupling between the tube isn't moving the whole fluid, which is then moving the fish. The tube moves. There's certainly eddies and so forth that are created that could potentially be sensed by lateral line and so forth. The tube which is the mechanosensory system of fish. The tube moves, and then the fish has to actively change its motor pattern to move in response to the tube. So the viscosity and the coupling mechanically between the tube and the fish aren't really, I don't think, relevant. Oh, absolutely, which is exactly why we've spent so much time trying to understand what's in the plant box. right? So, and I agree, like, you want to open up that loop, and actually our first paper in this was a dumb model of a plant where we did the closed loop system identification, and then we kind of opened up the loop, theoretically opened up the loop. I absolutely agree. In fact, that's why, like, Tim Keeble's approach of doing joint input-output identification, where you do both mechanical and sensory perturbations, and then you can sort of start to divide out the blocks, is essential.
In the very back of the room.
I'm just curious, thought about doing this with blind cave fish on your last experiment?
With what kind of fish?
Yeah, blind fish that live in caves.
Blind cave fish.
What do you
People have
think
suggested that before. I don't know enough about them to know whether or not they would have a behavior that would be as easy to elicit as this, but that's actually a good question. They're, as I recall, quite well adapted in terms of their mechanosensory lateral line. Getting them to track, therefore, might be a little bit challenging, but it's a good idea. I mean, I haven't tried it, but it's a good idea.
Question here.
In your control diagram, how to deal with the muscles activation function during the locomotion?
Well, we haven't yet, although we've tried. So we want to measure the EMGs and then at least correlate that with kinematics. It's been extremely hard. They have incredibly small muscles that activate, that are connected to 100 or so of these little rays. And the surgery has just been technically difficult. I think we'll get it. But the surgery has been technically difficult to do the EMG recordings of postdoc research scientists in my lab have been trying to do that.
Sort of along the same lines as the cavefish thing, I'm wondering how good the visual system on these things are if you can get different kinds of behavior if you can like high contact technical or these kind of things.
Yeah, great question. So the visual system is certainly pretty good. It's not as good as ours, but it's plenty good to see. We know that because when we move just the lights, they'll track them, and there's absolutely no electrosensory cue. In other words, when we turn off all the lights, and we keep the physical shuttle stationary, and we move the stripes on the tube, they will track it. So there's clear evidence that we get a strong spectral peak at the stimulus amplitude of a purely moving visual stimulus. But exactly how good it is going to be one of the things that we're going to have to, investigate in terms of contrast of vision. And
So when you're doing the sensory weighting of if you change the contrast of the visual stimulus.
Absolutely, that's a great idea. And in fact, this change of conductivity was a pain in the neck because you basically have to do wholesale water changes to decrease conductivity. And if you want any semblance of randomization, that means you're changing tanks a lot, and it's a pain in the neck. Now that we've kind of established that, we're just going to do visual changes of the visual system because it's a lot easier. We'll still do conductivity when we have to. But.
Okay, one more question.
So my question is about what's going on with the electrosensory system and how that's being used to detect the motion too, and whether self-generated motion could be, possibly changing the accuracy of the information about tube motion, and that's why they're moving down.
Exactly what we're trying to address electrophysiologically. So Eric Fortune, my collaborator, has done years of work on direction-selective neurons in the midbrains of these fish. So direction selectivity, Hubel and Wiesel got a Nobel Prize for looking at direction selectivity in, I think, cat visual cortex. So you can basically move some stripes and measure a particular neuron in the brain. And as you move the stripes, you get, and then in the other direction, you get nothing. And then, well, so he studied that for a long time. And we looked at how we're looking at how these ancillary, so if we look at the same kinds of neurons and then move a broad field stimulus while we're measuring in the brain, and we move it, say, at low frequencies, and then we add these ancillary movements on top, how do the coding properties of the individual neurons change as a function of the addition of this ancillary movement? So I didn't present any of that data, but that's something that we're hotly working on. Great question.
All right, thanks, Noah, again.